Good morning, everyone. I'm John Kramer. I'm the president of the Hudson River Foundation, and it's my great pleasure to welcome you today to the second Edward A. Ames Seminar of 2023. Hudson River Foundation has been running seminars like these for over 30 years and giving voice to scientists who will provide us with information on pressing issues of interest and concern up and down the Hudson estuary. And really to begin to have a dialogue that will allow us not just to understand these issues, but hopefully understand how the use of science can improve decision-making around things as important as plastics. Microplastics are not just important in the Hudson, it's a worldwide issue. And we're looking forward to hearing from our seminar speaker today. So with that, I'll turn the stage over to Jim Lodge, who will introduce our speaker.
Great. Thank you, John, and good morning, everyone, again. I'm Jim Lodge. I'm Senior Scientist at Hudson River Foundation. And before I introduce today's speaker, I just want to go over a few logistics for our webinar. Those of you who've been on our webinars before, this will be familiar, but we're asking folks to put questions for Beizhan in the chat, in the Q&A tab. So happy to have you guys use the chat function for communication with each others and providing additional information around about microplastics or other topics. But if you have specific questions, again, please focus those on the QA tab. And Beizhan's talk will go about an hour, and then after that, we will turn over to the questions, and I'll sort of try to organize those and pose those to Beizhan at the end. The other thing I want to mention is there's a closed caption feature, which is enabled in the webinar. There should be a button on the bottom of your screen if you want to utilize that. And so with that, I'm pleased to introduce today's speaker, Dr. Beizhan Yan. Dr. Yan received his PhD from RPI in 2014, I believe. And his advisor was Richard Bopp, who is well known to the foundation and probably many of you on this call. So Beizhan is currently a research professor at Lamont-Doherty Observatory at Columbia, and he's a leading researcher, not just contaminants that he did his PhD on, but also now focusing on the transport and transformations of microplastics and nanoplastics. The title of today's talk is Distribution of Microplastics and Associated Pathogens and Pollutants in New York City Waterways. Please join me in welcoming Dr. Yan.
Thank you. Can you hear me clearly?
Yeah, sounds great.
Okay, so let me bring up my talk. Just a minute. So I can share the screen. Can you see my screen very clearly?
Looks great.
Okay, let me close some of the, yeah, okay. So thank you, Jim, and thanks to invite me to give a talk at the Hudson River Foundation. As Jim mentioned, I'm, a graduate from the Rensselaer Polytechnic Institute, and my advisor is Richard Bopp, and my PhD work was supported by the Hudson River Foundation as well. So that's why Hudson River Foundation is supporting my career, significantly. So this is the work that I have done in the past probably 5 years, and this is not only by myself, but also, might have a lot of the collaborators, and including Joachim Goes, who is my colleague here at Lamont-Doherty, and also Philip Orton, who is from the Stevens Institute of Technologies, and also the Dr. Benny Commerce from the Dominican Universities. See, okay, so I'm trying to let me get the user personal view, use the point of views, laser point of views. Okay, so I will first to introduce the plastic pollutions and also what is microplastic as well. And then I will talk about the microplastic distribution in New York City waterways and beaches. Then I will talk about the in situ experiments I did, and to investigate the potential role of the microplastic as vectors for transporting pollutants and passengers. And then we talk about the microplastics if they are using the model to figure out how long they're going to stay in the New York harbors and where they ended. And then we'll come up some of the mentioning some of the potential solutions that we are right now is doing. And if I have time, I will introduce currently the network that I recently established, which is the plastic pollution analysis and sustainable solutions. We have a bi-weekly seminar as well in this network. So plastic was invented in 1907. The first one was bakelite, which is by the like a Leo Baekeland, who's actually invented in the Yonkers at the New York City. And later then he became a researcher at the Columbia University. And after the 50 years after that, there was a lot of the plastic being invented, including the polystyrene, polyester, PVCs, and polyethylenes, nylons, polycarbonate, polypropylene. You can see there are very different molecular structures. And these different molecular structures, which is giving them also different of the physical chemical characteristics of these plastics. So the versatility was one of the reasons that plastic industry really liked the plastics because of the molecular structure. So they are different of the density. You can see some of them goes to the 0.9 and some can goes to 1.6. However, majority of them was actually below 1.2. So among these plastics, the polyethylene and also polypropylene are two largest plastic being produced. And so in the Gaia et al. In 2011, he used the data and then from the 1950 to the 2015, and he calculated that the plastic was produced globally was 8.3 billion metric tons of plastic being produced, and 1/3 was still in use in infrastructures like in the PVC pipe and also the buildings, etc. 2/3 was actually discarded into the environments. And so among that, some of them was pathetically was only 10% get recycled. So to me, I think our research should be focused on how we can improve the recycle rates, and also so that they mean less of the pollutants can infecting the environments. So microplastics was when they are discarded into the environments, these plastics can undergoing the physical, chemical, and biological weathering, and then they became became smaller and smaller, and then eventually can reach to the nanoplastic, which is nanoplastic is less than 100 nanometers. And the microplastic definition is basically from the 100 nanometers to the 5 millimeters in that range. And so also that not only have these secondary sources, but also you have primary sources. For example, some industrial pellets that are being used as precursors was also in that range. And also that another one that microbeads which is around a sapphire one, that are using as a scrubbing, agent that has been used for the Cosmo products has been widely used. And New York State has tried to, ban the usage of microplastic in those, and was still not successful, but New York State did a great job to ban the single-use plastic bags. So at the Lamont, our studying the microplastic was about 6 years ago, which is actually from high school program, which is a Lamont Secondary School Field Research and Progress program. And this program is basically high school summer intern program. And so the teachers who is Deborah, who is a teacher at the time that she realized the importance of the microplastics and find out all the news and medias, so that she wanted team to do some research. What the first steps is actually to go to the grocery store and then digesting and then to find out what actually in the tissues of those microplastic in those shrimps, fish, and oyster and clams. And not surprisingly, in each individual shrimps and fish, they are find microplastics. and find out that you can have microfibers and can have like like rounded like microbeads, etc. And so then they also confirmed that using that FTIR, find out the FTIR, which is the Fourier transform infrared spectroscope, to find out the fibers that they find out is actually is PET, basically is polyester. And these polyesters, basically coming out from the laundries, from the like blanket, from a lot of the carpet, etc., from the home usage as well. So so this is give us some of the in momentums. We know that the microplastic was the issues in the food, so we want to see whether they're actually in the New York City harbors and the waters. So that we starting to collaborating with the riverkeeper. And so that John was very nicely to providing us the boating time so that we can collecting the samples along the lower Hudson and also around the other like attributary like like East River and the Harlem River and also the like Long Island Sound, etc. So that was actually starting off the research. And so with the Hudson River Foundation support, we continuing to do more work in the 2019 and 2021, and we collected like water samples from. So after basically we collect in the several sites. One is close to the Eaglewood, and then one is the sites we collected from the like North River, Westward Trim Plants, which is the main like Hudson, and then Pier 25, and then that lower Upper Bay, and also the Newtown Creek, and some sites in the Harlem River, etc. So we basically collecting the samples, water samples. After that, we filtered, and then we remove the organics through using the hydrogen peroxide, and then we use NIRAD and FTIR and Raman to identify the particles that we identified. And you can see that in one of these, you can see that was a microbeads right there, and we also see quite a lot of these other particles like black carbon, which you can clearly see in this plots here. So you can see that the microplastic was at relatively low before they are getting into the New York City waterways, and you can clearly see there was an increase in the New York City waterways, especially high in the Newtown Creek, which is a long narrow inlets that is 3.5 miles, and which is recent, like 2010, they are claimed they're being like announced as Superfund sites. And clearly there was Hudson River was very high in all those in the Newtown Creek sites as well. So this is clearly giving us some of the like thought about microplastics, which is the potential source, and we think probably CSO and also the wastewater treatment plants discharge potentially was the major sources. The reason we're saying that is we not try to blame the wastewater treatment plants. I know that the wastewater treatment plants is that they did a good job to remove the like pollutants that are coming out from the person we living in the cities. So they're just bypass, and then they are not designed for microplastics. Currently the wastewater treatment plants was not designed for that. So that's why they are coming out. So also that we also confirm that some particles like this is Raman techniques used that we can confirm the particles about like 30 micrometers that is PE and like polypropylene. And so this is things happening in both like Newtown Creek and also in the Pier 25 in most of the samples we can detect it in the PE, PP, and also polyesters as well. So we know that it's not like the microplastics is in the New York City waters, but the situation is what is actually for the people get exposed, what scenarios that could be mostly get exposed. The New York City drinking water, they're coming out from the upstate, like reservoirs. And so they are basically, like not in the New York City waterways. And so, and you may get exposed by the, like wave breaking in the, along the coastline, but the most of the place that was not really get exposed from that. Of course, you can get some food, but there was also, that's really, like rare conditions. So, but one way is actually being get exposed is actually from the beach. So then this summer, we actually have the high school students, and then we, starting collecting the beach samples from the 6 sites which is from the Orchard Beach, from the Flushing Bay, from Pier 4 beaches, and also from the Coney Island and the Rockaway beaches. So in those places, we actually collecting the, samples, and not only that, a beach sample, we actually separating the beach sample into two locations. One is low tide, one is high tide locations. The reason is because the high tide is people who typically sit and then enjoy, and then low tide, which is basically the person that play in the water. And so we want to see whether they have the whether the dynamic changes causing the microplastic distributions in those sites. So then we collect. You can see that we're putting a roll, and then we actually collecting about like 9 samples, and then we mixed it, and then we bring back and then do the quite a lot of the analysis. And so, and we basically try to compare the low tide and high tide conditions. So we also not only collecting the samples, we also beach sand samples, but also collecting the plastic waste in those sites. So we basically cover an area about 500 square feet, and then we collecting areas plastic waste we saw in this 500 square feet area. And you can clearly see that it was some sites like a Pier 4 beach. It was pretty, filthy with with a lot of the plastic pollutants, which is basically more than 5 millimeters. So we call them macro plastic, but they are doing quite a lot. You can see there was a lot of plastic cap, and also the straws, etc., and pens, and all things that you can see that, and also fragmentations of the plastic as well. So, and also in the Orchard Beach, which is quite, crowded when we're doing the collections, and we see quite a lot of the plastics as well in these sites. So we also, that's the reason we try to see, we try to compare the high tide to the low tide conditions to see what's actually there. And it's clearly showing, this is in Orchard, that you can see that it was, this is showing the grain size of the sand that we collected, and you can see the grain size was much higher in the high tide than the low tide, which is reasonable because that's basically controlled by the wave. And so we also see quite a lot of these, plastics that, like for example, cluster of the plastics, and also we see the fibers, and also we see some of the micro, like maybe even nanoplastics, and also we see some of the, like fragmentations of the plastics in there, and some of them being identified as polymers. Some of them polyester, some of them, PE and PP, polyethylenes, etc. So then we compare the low tide and high tide situations, and then this is Coney Island. We can clearly see the Coney Island high tide have the much higher concentrations on the number of the microplastic finding in grams of the sand, and you can see there was much higher. And then the low tide, located the lines. And so this is not only found by us. So we actually also measure, because one, we also get the sample from the Puerto Rico, the beaches from there in Battery and the Maya beaches. You can see they are consistently showing much higher of the microplastics in the high tide than the low tide positions, also similar in these cases. So then I have connections with the other researchers in Europe and also China. We actually consistently showing that the high tide in the high tide, higher a microplastic concentration in the high tide than low tide. So this is basically showing that this is not only in the New York City, but it's all places. So one potential reason, which is I came up, this is called bathtub rings hypothesis. The reason why I'm saying that is because that we think about the bathtub rings, which is basically you bring in all these floating materials from the the dirt and the soap and oils that have been coating on the highest levels, which means their lower levels being bring up to the high levels. And this is probably analogs to the whatever situation happening in these high tide, low tide distributions. So basically during the high tide conditions, so the low tide lines is under the water, and this is energetic. So basically the microplastics in the low tide lines was being resuspended. And when they are doing resuspensions, they are being pushed by the wave, goes to the high tide lines, and then some of them being trapped in there. And this is continuous doing this, moving. So that's why we see a high tide condition. So it's a very, simple, explanation, but actually can be very meaningful. The reason is because we have limited resource when we try to, try to find out where we basically collecting the, finding the microplastics. So basically, you can see that we can probably go to the high tide locations, and then we can do pick up these plastic pollutants there, and then which is more efficient so that we can, and then with time, probably a low tide place to also continuously transport some of the plastic back into the high tide condition, sites. So which is I think this is giving us if we want to control the plastics pollutions in the beach, we probably can try this way to, if the resources is very limited, we can potentially using culturing. Basically, is actually collecting or control that what is actually plastic pollution in the high tide locations. So this is basically research that showing this can be used for this purpose. However, we're not really surprised for the microplastics in the New York City waters. As Jim mentioned, they are they are all over the place. They are deep in the oceans. They are up in the mountains, and also they are even in the Antarctica, and also sea ice in the ice thick, Antarctica, and also in the polar north and Arctic regions. So we know they are there in the environments, but the question is, so what? So one question is that they actually carry the various pollutants. As we know that the New York City's Hudson and New York City waterways is not famous for the microplastics. It's famous for many other pollutants like the PCBs and PBDs, and also the other pollutants like metals and plus also the passengers. So that's why we think about we could potentially see whether they are actually, when the microplastic being released in environments coexisting with other pollutants, become vectors to carry them. So that's why we're starting to do this research about 4 years ago or 5 years ago. So there was in order to study whether they carry pollutants, we can have two options. One option is we can collecting larger volumes of waters, and then collecting microplastic in the larger volume of waters, and then picked up those microplastics, and then do analysis to see whether they are contain some of the pollutants. However, this is very difficult to do that. There are two reasons. One is that you need a very large volumes of the water to filter to get the really microplastics and also nanoplastics. It's almost impossible. You need probably more than 10 cubic meters of water to really get enough. And also another thing is actually, if you try, you may study the microbial communities on these pollutants on this microplastic surface, but when you try to separating microplastics from the other minerals, other particles in the water, you not only have microplastic in the water, you have a black carbon, you have the clay, you have the sand, all these tiny particles are coexistent. If you try to pick up the microplastic from that, you may use gravity separation using sodium iodide or sodium chloride. Why you do that? You actually interferes as soft pollutants. So that is kind of difficult to really study that. So our team come up with this solution, that is we can use called this in situ experiments, which is actually the idea coming at the starting point was from Deborah, that she actually be the first author of one paper. So basically, we can get these pellets, and then we put in all these pellets inside of those T-ball. This is stainless steel ball, and putting inside of stainless steel, we sealed it, and then putting inside of the a basket, this is galvanized basket. So then we basically putting this basket below the water, and then over a certain time, and see after a certain time, we bring back and then see what's actually up to the surface. So this is basically the work that we did first. And so you can see we actually putting we can about 9 or 10 of these different type of the particles, and we put it inside of this T-ball and then put it inside of this basket. Okay. And then we put in there into the several sites. That is we selected 6 sites, or from the Eaglewood Cliff, and also the North River Westward Trim Plants sites, and also the site at Pier 25, and also the sites Newtown Creek and Flushing Bay, and also the site 6 is Harlem River. So you can see that we actually calculate on the depths of the water during the time, and so we know what is the lower tide conditions, and then so we see the depths of the chain, so that we put in the basket, it was below the lower tide conditions, so that it was all during the weeks of times that the basket will always stay under the water. So that is basically the way we do have a lot of the try to figure out what is actually the depths of that we do. So we actually put in there, and then 3 weeks later we go back and then we retrieve it. And that day was pretty depressing. The reason is because not because of weather, but because the first 3 sites along the Hudson, we actually could not find it. It disappeared. So all this basket we deployed it there 3 weeks before and was not there anymore. And it was very depressing. And and then fortunately we go to the other 3 sites and we got back, we got from Newtown Creek, from Flushing Bay, we also from the Harlem River. And then in the samples we got back, we see that it was actually they're very rusty. The reason that we actually using this called galvanized, steel, which is they are not very good, not very good to resist the saline waters. So they get very rusty. And also another reason get lost is because they are in the mainstreams Hudson, and so the water flow very quickly. So that basically they are carry the basket and the bank to the pillar that we are putting in there. So which is because of rusty, because of the bang continuously, hit the a pillar. So then that's probably the basket get lose integrity and get lost. I'm pretty sure that they are probably still somewhere in the in the sand of or sediments in the Hudson River. So or maybe even goes to the lower upper bay or lower bay. So fortunately, we have these 3 sites with 3 samples came back. And in those sites, we also measure the pharmaceuticals in those waters. And so it's so we measured the 3 different pharmaceuticals. We will give the 3 examples like atenolol and also ibuprofen and also these compounds as well. So we measure those compounds in the water samples by itself. And we find out, of course, this is not some other research, Riverkeeper and also the other research also showing similar like that. You have a higher concentrations of the pharmaceuticals when they are close to the westward trim plants like North River, Newtown Creek, and the Flushing Bay. And we can see them a high level into in there. And this, as I mentioned, this is not a new. And so then we also measured what it actually go on the surface of these pellets. And so then we calculate on this called pathogen coefficient, which is between the solid and the water, phases. And so they are, so basically, we, get the concentrations of these pharmaceuticals on the normalized to the surface area of each individual plastics, and then divide by the concentration in the water. And so it's very interesting that we can see they are pretty much similar. So they are very, very much similar, concentrations, the coefficient. So this is give us a hard time to understand. So, but then we realize that, when, especially in the second day when I deployed it, I go back and then check it and I find out the green color coating on the surface of the pellets. And clearly that, like a green color is because of biofilm, and this biofilm was colonized the surface quickly in days. So when we think about it, when we deploy this one in the New York waters, because New York water was very enriched with the nutrients, so they encourage on the surface on the colonization of bacteria, of archaea, etc. And then you basically, this partitioning is not between the water and the plastic, it's basically between the water and the biofilm on the surface of the plastic. So that's why we think is because we get this very similar ratio, similar levels on that, okay? So, but this work has three basically, like limitations. One is that because that work was done by, by using my own money. And so we are, try to find an inexpensive way. So that's why we get this galvanized, like basket. And so it's $20 for each piece that which I can afford. And so, but we, as you know, that because they are get rusty quickly, so that's not really good. So we need to use, like expensive stainless steel, like a box or a basket. The second, issues of this study is that we actually putting the microplastics in the T-ball. And then when you put in the T-ball itself forming an environment that by falling, there was a lot of bacteria or whatever growing on top of the, like on the surface of the T-ball, basically is forming its biofilm that is actually hindering the water flowing through. So which is a problem because that is actually hindering the exchange of the water with the, plastics. Another thing is also we put in there 3 weeks, we didn't go back and then we lost the samples. We need to find some place that we can have someone can look over for us and then monitoring that. So then we, coming up, we got this supporting from the Hudson River Foundation, which is very nice so that we can get the expensive, like a stainless steel box, which is each of them cost about $500. And then we also selected 2 sites that is, we know that we can, our sampler can be, can be in good hands. One is the Brooklyn Navy Yard. And then in there that, we have the collaborations with the Billings Oyster Project. So they are helping us to, like keep an eye on that. And also we put in one in the Newtown Creek, and the Newtown Creek Alliance was also helping us to monitoring the sites. And so, so then we did, deploy these 2 sites, and in each site we deploy the HDPE and PVC pellets. And so we have the like a 9 replicates rather than the like a previous one we only have 1. Now we have 9 replicates for each site, and so we basically have like HDPE pellets. This time we're not putting in the T-ball. We actually dig a hole across the dig a hole like right in the center, so that the pinch a hole in each pellet. And then we string using the stainless steel strings. And then we tied these strings into the basket wall, the wall of the basket, like stainless basket, so that we can, like a tight, very tight in there, so that there was no hindrance of the T-ball, which is, which is very, like that, preventing the by falling a lot. And so questions is the good thing is of that one is that it's really can losing the like interference of the biofouling, but the bad thing is actually that it takes a lot of time and patience to dig a hole through this small tiny like only like 2 to 4 millimeters of the pellets. Basically is 1/16 to 1/8 of the pellets that you dig a hole right in the middle and then string that, which is I'm pretty sure my lab like technician doesn't like that job at all. Takes her like 5 months to dig a like a 5. Basically, we need a 2,000 pellets, and for 1,000 for HDPE, 1,000 for PVC, and it took her like 5 months to do the work. And I appreciate that she's so patient to do the work. I cannot do that. And then we actually deploy this one into these 2 sites, and with the help from my student Federica. And so, and then 100, and actually 100 days later, we bring back, but during these 100 days, we actually visit there every week. And so that we poured up, and then we basically clean using the silicone, like brushes to clean the, like the basket and also clean the surface of the pellets so that to preventing them by falling. So this is basically we did using quite a lot of efforts to do that work. And after that, we measure the PCBs and PBDEs, and also we measure bacteria community using the 16S rRNA, and which is like made by the Dr. Bernie Connors from the Dominican University. And we're still doing the metals analysis. So this is basically the results for the PCBs. And I know that the Hudson River is very famous for the PCBs. And so has foundation was, so I'm pretty sure audience really know what's actually PCBs there. So so we measure those basically water samples first. So basically we give it the 1 liter of the water in the filter, and then we measure the PCBs on the particles. And you can clearly see this is on the top one here. You can see the Newtown Creek have a much higher level than the Navy Yard, which is reasonable. And we can see there was a little bit difference in terms of the PCB distributions among different of the PCBs compounds with different the chlorine, number of the chlorines on the PCBs. And so we also calculate on the concentrations, that is the PCBs concentrations in per grams of the system particles. This is basically in the red like dot here. This is the first two of the like a column. This is talking about the water. And the second, in the middle of two plots, which is the top one, is the concentrations in the HDPE, the plastics. And so we actually separate into three. One is in virgin, we basically is a clean one, and so without putting into the field. This then we actually have the samples from Newtown Creek and from the Navy Yard. This is very interesting results showing that of course that the Newtown Creek, when they're putting Newtown Creek and the Navy Yard, you can see a higher level in the virgin pellets, which means that they are coming out from the environments. And also we can see there are also Newtown Creek have a much higher level in the Navy Yard, which is like as reflecting in their previous environments, which makes sense. And we're also seeing that in the HDPE, like plastic, we can see much higher level, which is 10 times higher than in the suspended particles collected from the water. This is actually, showing that our plastic, HDPE plastic, is actually can concentrate the PCBs from the waters, because it's much higher than the sediments, the suspended particles. So which is clearly showing they are HDPE can concentrate the like a plastic onto the surface. How about the PVCs? We can see they was also being affected by the environments, the higher level, and we also see consistently high level of in the PVC in the Newtown Creek, also in the Navy Yard. However, we see a much less of the concentrations compared with the suspended particles. So we see that the PVC can absorb on the surface on the PCB compounds. However, they cannot concentrate as HDPE. This is weird. So we still try to figure out what's the reason for why we have such a like distribution difference between the HDPE and the PVC. One potential reason is because the HDPE, the molecular structures is just at least like a carbon hydrogen, like an alkane, like like a diesel. So basically, HDPE surface very hydrophobic. So basically, and also PCB is hydrophobic. So they are coming together. So that's potentially one reason with the enrichment of the HDPE on the surface. PVC contains chlorine compound, affecting their hydrophobicity. So that's potential reason leading to the less of the concentration on the surface. Of course, we are still doing the research. We try to figure out what's the reason as well. So how about the other one, like a PBDE? The PBDE is the bromide compounds, and so they have a molecular structure like this one here, and with the ester oxygen here. And so they are being used for as that the PBD is basically flame retardants. And we still see the same thing, same patterns that we have a higher in the Newtown Creek than the Navy Yard. However, that in the HTPE, we also see the similar patterns that we can see that the samples we collected, putting on this pellets, putting on the sites was a higher level, and so clearly showing that that's the some of the other absorptions. However, the concentrations of in the HTPE was not as high as the system particles. So which means they are absorbed to the surface, but it was not a concentrate on the surface HTPE. And also the PVC, even they are not concentrated, they are actually, the virgin has enough of the PBDE, which they are used inside as a plasticizer. So that's why we have a relatively high in the PVC and of the PBDE concentrations. So we are, reason we are think about why the HTPE this time didn't concentrate the PBDE is because the molecular structures of the PBDE, you can see they have this oxygen ester, function groups there. And this oxygen can increase the polarity of the compounds and leading to potentially to have less of the, absorption to the surface, okay? So this is basically, our pollutant studies. So then I collaborating with Connors from Professor Connor from the Dominican College, and then she's she's actually using get our samples, and so she got basically we give her the water samples and then 750 samples, and she filtered through the filter, and then using extract the DNA from the filters. And then she actually separating the bead samples that in two categories. One is the total DNA extract from the beads. So basically is the extract everything from the beads. And then figure out what's the DNA extractions, figure out the microbial communities. Or they are, and also she actually putting that beads and then in the shaking table, and shake in the saline water for 30 minutes at room temperature. And then she basically measure out the PCB in the species. So basically is to try to see what is actually loosely attached microbes on the beads. So basically this is total DNA extract. This is basically microbes that are loosely attached, so potentially can get lost during the transport. So then she used that the regular, all this, she sent the sample to do the, PCR work, and then she did some work on the bioinformatics for the DNA structures and just figured out what's the actual microbial community. Okay, so this is basically a sample, a plot showing the results, and it's quite busy. However, that I can walk through you. So basically, we select the 5 taxa, in the microbacteria, natural, superlithus, and also the others. So the microbacteria, which is the a group of bacteria that is actually coming out from the human disease, for example, like tuberculosis. And so this is very bad. And also natural, this second one, natural superlithus, which is, basically is a marine archaea. This marine archaea, they are actually oxidizing ammonia to nitrate. So in the well-rich, nitrogen-rich environments, these bacteria will grow. And also we also, measured the, Mesococcus. And this is basically the bacteria that actually can use methane and for their energy and also for carbon sources. And so that's basically is when there was a lot of methane in the environments, then it's trouble. Another, a first one is bacterioloids, which is commonly found in the human guts. And also another one is a Helicobacter. And a Helicobacter is basically in the digestive tracts in the variety of the animal species, including the birds and also including others, like human, but also a lot of birds. So we did actually in these locations we find the goose and also the ducks and other birds while we are doing the experiments in the sites. So The plots here showing that the relative abundance of these 5 taxa, and in different samples. These water samples, which is Newtown Creek water and also Navy Yard water samples. You can see they are pretty similar, and then they're in both in Newtown Creek and Navy Yard. We can see bacteria coming out from the human diseases, like from the tuberculosis. This is pretty bad to me, so that's why I washed my hands after each time after we pick up the basket. I always using the alcohol to clean my hands. So, but this is not good news. And so also there are also contains the Helicobacter, like group of the bacteria, which is from the animal, like from the bird, from something like we do when we collecting doing the samples there, we do find out like a seabird and also see some of the duck there as well. So this is clearly showing there are a dominance in these two. We also see some of the like human guts and also from others as well. So this is basically 2 water samples. They are pretty similar. However, the plastic, when we deploy in the environments, they are very different for the plastics. So this is from the 4 samples from the Brooklyn. This is 4 samples from the Newtown Creek, and you can see that for example, for Navy Yard, we can still see on the surface of the particles. There are some of them are loosely, but also they are tightly also as well. In the on the surface, we can see this microbacteria. This microbacteria, as I mentioned to you, is the human disease bacteria, and so basically the plastic is carrying them as well, on the surface. And this is surprising to me as well. The second one is actually this mesotrophic bacteria, which is growing in a huge amount there as well. So this is probably reflecting that the environments that we are in the Navy Yard. That when I was deploying my sample, I do see air bubbling from the bottom. So they may potentially have sites that the sites was actually filled up with methane. We didn't measure methane in the water samples. We should do it, and probably next time we should go back and then collecting samples, measure methane in the water. However, this is clearly showing the mesotrophic bacteria was actually dominant. And also we see the bird, one, but for human guts material, we didn't actually see too much in the Brooklyn Navy Yard. Newtown Creek is completely different. Even the water, showing the similar, microbial community, and we can clearly see Newtown Creek. The samples contains a highly relative abundance of the microbacteria, which is from the human diseases. But we also see the, microbial community that is enriched with the, this marine archaea. And they are basically showing that it's a dominant one over there. And as you know, Newtown Creek and ammonia was probably also one major source coming out from the sewage. The reason is because sewage in the it's a byproduct. Ammonia is a byproduct of the decomposition of organic matters in the sewage plants, and so this is kind of released into the environment, and also and we already know that Newtown Creek is pretty filthy in almost in everything, and so but clearly showing the nutrients. This is actually use the nutrients to microbial distributions of that. We also see other one, but it's much relatively small, abundance. So this is actually very interesting. So the basically 2 interesting things. One is that the bacteria, the water samples contains very different, a microbial community compared with the plastics. So this is actually a plot using basically same information, but we putting into called the principal coordinate analysis, PCOA analysis. She tried to visualize the similarity between different groups, and you can see the water two water were grouped together, and then Navy Yard grouped together and Newtown Creek grouped together. So this gives us 2 informations. One is that plastics. The surface of plastic microbial community are different very different from the waters. This is not, like the other study, like one study from the Woods Hole Oceanography Institute, also showing the same story, but they are using like the marine debris. And they find out the microplastics, plastic surface have a very different of the micro communities than the waters, which is makes sense. One part of particular, things is that showing that the plastics has its own, community, and that's why the term comes like plastisphere. So that's mentioning this is not only finding in us, but also in finding in other studies as well. Another thing is also showing that plastics, a bacteria community clustered by the size, not by the plastic type. So you can see the new navy yard is different, very different, but the HTPE and the PVC are very similar. So they are basically very similar. And Newtown Creek also showing different, but they are HTPE and the PVC plus are basically the same. So this is kind of giving us that showing the conclusion is that the bacterial diversity clustered by the sites, but not by the plastic type. So I'm giving you a little bit of sub-summary. One is the wide distributing of microplastic in New York City waters that we need to really control and remove efforts. Another thing is plastic can serve as vectors for the transport of the pharmaceuticals, and the plastics, especially HDPE, and also maybe other like LDPE as well, like low density polyethylene as well, probably can concentrate the PCBs from New York City waters and not the PVC. However, more studies still needed. Another thing is the passengers can both loosely and tightly soft to the plastic surface. And we find out different micro community on the plastic surface than in the water, which is plastisphere, which is a term like plastisphere as well. Another thing that we mentioned is the bacteria diversity clustered by size, not by plastic type. So basically this is whatever the research we showing in the passengers and also the pollutants on the surface. But the questions we are trying to also answer is that how, if there are plastic particles, how long they're going to stay in the like New York City, this New York Harbor regions, and where they ended. So that's why it comes to this work by the field orator, and he's also in the call today. So the method he used is actually called Stevens ECOM, which is estuarine and coastal ocean model. And then basically using this model, we have 4 assumptions. One is actually inject the like 1 kilogram per second of the, here is microplastics particles, tracers through the 2 sides. And then this is released from the bottom. And then the 4th assumption is neutron buoyancy particles. And this is not very far away from the real situations, because I mentioned to you, the density of the microplastics basically is close to the water. And so they are basically floating, and most of them particles are less than 1.2. So which is not bad in assumption. And also they are released over like 12.4 hours that they can capture the average effects across all tide phases. So, and then that, like we keep putting the 4 scenarios. One is 2 tide, one is neap, and one is spring tide release, and also putting in 2 stream flow conditions. One is low, one is mean. We didn't do the high, flow conditions. That's really because that's, for one reason is that the high tide was not really, high tide was not really, rare, often situations with relatively rare situations. So that's why we go with the low and mean conditions. And then residence time, we have a definition that is the time for the mass here is basically is the, microplastics mass in the harbor regions to decrease by the whole harbor regions and to decrease by 63%. So basically it's reached to the 37% level. And this is a definition of the resident time. This is definition has been well accepted and in the modeling and also by the, field works as well. So this is a basic model. And so the harbor regions, the define is from the north to the harvester, south to the, Atlantic Ocean. And so including the Newark Bay and it's part of the Long Island Sound and also Jamaica Bay. So basically cover the area both inside of this box, red box. We putting two release sites. One is in the North River, the discharge sites on the North River treatment plants. One is from the Newtown Creek, where the Newtown Creek wastewater treatment plants release or discharge their pollutants. So we have basically these two sites to release. Okay, so this is animations about when they are released, the particles coming out from the North River right in here. But in the two conditions, one is the nip tide, one is the spring tide release. And you can see that was they are when there was nip tide, because that was a very weak, relatively weak tide, and the nip tide conditions, they are water can stratified, and so the water stratified well, and then so that some of the microplastic released from the bottom was being kept by the deep flow of the saline water, and then they are moving up to the upstreams and even reach to the Harvester and also Tabanzi area. So that's one scenario. Nip tide. For the spring tide, they are basically have less, but it's have much more, goes down to the north New Jersey coastlines, because they are spring tide has the more vertical mixing. So this is just a snapshot of the using the North River releasing sites. So have a nip tide in one day, and you can see there was from the releasing from this new North River sites, they're going fill up in the lower bay, and also goes to the up. So from the up bay, and it goes up as well. So, but also that you can see the spring tide, because there have the more mixing. So there are a lot was actually coming down to the more into the lower bay, and some even past getting into the Atlantic Ocean. This is the day 5 scenarios, and you can see there was the major a lot of the microplastic was being trapped in the in this lower Hudson, and some of them was also come up. But compared with the spring tide, more was coming down into the North into these shorelines of New Jersey, and some will actually end into the Long Island Sound as well. But it's much less concentrations compared with in the Hudson River compared with nip tide conditions. So this is basically the that research that is showing that in the releasing from North River, if we releasing this from the two sites, one is goes to the we know that one is goes to the the sites at North River wastewater treatment plants, one is goes to Newtown Creek sites. You can see if we're releasing from the main Hudson, then they are being trapped into the a lower Hudson, and much more we trapped, and then less getting into the Long Island Sound, and also less getting into the relatively less getting into the North River, New Jersey, shorelines. However, when they are released from the East River, you can see there was much more getting into the Long Island, and less getting into the Hudson River, lower Hudson River, but much more was actually getting into the New Jersey shorelines. So this is basically the animations, and I also do the snapshot, and the snapshot showing that in 1.5 days after that, you can see there was much more in the lower Hudson, but if we release from the East River, you can see there was more in the Long Island, in the west side of Long Island, also in the getting into the lower bay, getting inside into the Atlantic. And this is day 5, and releasing from two sites, and you can see there was still quite a lot was actually trapped in on the lower Hudson, and but for the East River, there was much more getting into the from the East River, getting into the Atlantic. Okay, and when there was 20 days later, and you can see there was pretty much similar, and but it still have a whole bunch was actually in the lower Hudson, and eventually within 33 days or 40 days, then most of the microplastic was actually moving out, getting into the oceans. And so either from the Long Island Sound, the majority was actually coming out from the like these morose channels, these sites as well. Okay. So then we actually calculated in different scenarios, and was mentioned we have two locations, discharge sites. One is North River, one is the East River. And then we have different scenarios, like we have a neap tide, but at the low, like a mean discharge of the water. And also there was a mean discharge, but in the spring tide conditions. And we also see that the neap tide in the low flow conditions and also low flow, but in spring tide. You can see the highest residence time was actually the one which is in the like a 30s, sorry, in this like 30s, like a 3 days, which is in the low flow conditions in the neap time, but it's actually is released in the North River. And you can see in the East River, it was much lower residence time. And the low, that the best, like dispersion, like scenario is the mean, like flow or high flow at the neap time and released from the East River. So you can see that when you, the particle from the, like from the different sites from different scenarios, but they are basically from 10 days to the like 33 days in that range. In this and resident time of that of them in the New York City waterways. Okay, so then basically the summary is that the Stevens Institute that this model was run, and then we find out the resident time for the microplastics are 11 to 33 days within the harbor region, and the longest time are for the microplastic releasing into the Hudson on the neap tide due to the, and also is actually along the Hudson River when we release from the Hudson. If the East River, it was actually much more quicker to be dispersed into the other regions. So the issues is that when you really have a dry period, which we have a lower flow, and then that's there's a time, and the microplastic can stay in the water in this like in the condition harbors for much longer time. It can go to like 33 days or even longer. Okay. So as I mentioned, that we have like this is kind of a study we did and in the past like 4 years. And so, but we also realized there was limitations. The first limitation is that we really lack of available data about the lower end of the particle size of the microplastic and also nanoplastics, especially for those nanoparticles or microplastic less than 20 micrometers. The reason is because the traditional method that is used was not be able to really identify and detect these microplastics in the less than 20 micrometers, no matter whether it's Raman or FTIR. So then the solution is that we have developed method with the collaborating with my colleague at the chemistry department to develop advanced Raman techniques. These Raman techniques that can really identify the particles down to like 200 nanometers, even to goes to 100 nanometers. And we have successfully identified these nanoparticles in the bottled water. And our recent research showing that in 1 liter of bottled water, we find more than 15,000 of the particles, nanoparticles in 1 liter of the water, bottled water we bought from the grocery stores, which is bad news. And but this method can be used for this, like relatively clean samples. However, for environmental samples, there was one challenge. The challenge is that in the environmental sample, especially this water coming out from the New York, there was tons of the coexist black carbon particles there. And black carbon basically is that they are accepted the Raman across the wavelength, so they are burned during the Raman identification. So the combustion of that interferes our method to do the work to measure the microplastic from environmental samples. And black carbon is difficult to remove. Recently, we actually developing the method, and we can successfully to remove the black carbon. So we will use that in our environmental samples including the samples from the Hudson River. So stay tuned. We potentially can showing you more advanced results in later time. Another shortcomings is that the representative water samples. We collect 4 liters of sample, which basically is a snapshot. And as you know, that the water samples, 4 liters was not representative. First thing. Second thing is also, it's for the larger and also smaller nanoparticles, they may have these spatial and also temporal changes. So in order to really get, like a representative samples, we need a larger volumes. And so that's why, but the problem of these larger volumes, the filtration is difficult because it's almost impossible to filter the, like 100 liters or, 1 cubic meter of the sample, 1 cubic liter of the samples, because, like filter, even just 1 liter will, especially we collect it from Newtown Creek, they will fill up the pore size will make it impossible to filter after 1 liter. So right now we're developing method that using advanced filtration techniques that to actually concentrate those samples quickly. Within 1 hour, we can, concentrate the 1,000 liter of samples, so that we can use that, and then we can get the much more representative of the water samples. So this is a current our, progress. And so we'll be done in probably in the next 2 or 3 months. We should be able to come up with this prototype, and then we can probably use that in the Piermont here, and also probably use some of the other places like Governor Island, etc. Another thing is also we identify these microbial activities. Basically, we didn't do the activity that we using the 16S RNA, our rRNA data. That is actually reflecting what is actually what kind of the microbial, bacteria there, but they didn't showing us what is actually doing, what they are doing. So if we try to see what's the microbial activities, for example, are they doing more like a, hydrophobic, like a hydrocarbon decompositions, or what kind of chemicals they're doing, what kind of, activity they're doing. We need a better, method, which is we need a shotgun metal transcriptomic, method. And so that right now we're actively looking for a small fund for paying for that analysis. That will help us really knowing what is actually the microbial doing in our, on this surface of the microplastics. Another thing is also that the model we are providing is, like a very coarse spatial resolutions, and also with, like a density which is close to 1, and it would be good actually to have more model support that can doing much better, other, finer resolutions and better density work as well. Okay. So this is basically what which we found in the environments, and also how we can solve the problem. Because this is not, like a nuisance. So one thing that we think about is we saw a lot of polyester coming out from this wastewater treatment plants, and also from the water samples we collected. So which means they are tied to the laundry. And also laundry waste was not only found in New York City, but in all over the place. In the oceans, there are about like 1/3 of the microplastics are coming out from the laundry. In here you can see. And so the laundries was, the waste coming out to the wastewater treatment plants, but as we mentioned, wastewater treatment is not designed to remove them. So, then, basically the 10%, rough idea, 10% getting into effluent, and 90% getting in the sludge, and this effluent was contaminated the water in the, in the oceans and also in the estuaries. Okay. So how we can solve the problems? So we have, I did some work using my home, in instruments, washers, and then we collected the wastewater from 3 pounds of the clothes washed, and then we collected all this wastewater from there, from this, one wash, and we can see quite a lot of these fibers come out, shed out from the, washers. And then, we are not only identify these fibers, but also we see a lot of these micro, plastics. And then we even see a lot in the nanoplastic, which you can see this is 0.6 microns. And however, we can see the particles less than 0.6 microns. That was basically millions of the particles that are coming out from the wastewater from the laundries. And this is the basic first time that we can identify these nanoplastics coming out from the laundries. And so right now I'm actually, leading a team that doing the research to developing these advanced filtration techniques, and also find out the closed loop solution to reduce the leakage of the synthetic fibers, microplastic and nanoplastic from the laundry. And you can see I have two enthusiastic students, Yanni and Siddhant. Siddhant is already graduated. And so we are developing the filtration method, and preliminary results showing that it's much, this is the filtered water. This is the water being retained as a sludge. You can see there was a much big difference, and we can, looks like the filtration works that looks like their works. Okay, so this is basically the work that we are currently doing, and we'll continue for another 3 years to do the research. To do the developing this method and then integrating that design into the washer machine and the dryer machines. And so this is a work that, as I mentioned, not only just by myself, but my close collaborators, and also from my lab work from the secondary school, and also from my students, from the from PhD students, and also graduate students as well, undergraduate students as well. And also, I'm especially thank the Hudson River Foundation providing and my support, and I mentioned Hudson River Foundation is very important, significant, supporting my research because they are supporting my PhD work as well. So after PhD, they are also supporting my research at Lamont-Doherty. And also this work also gets supporting from Billings Oyster Project, River Keeper, and also Newtown Creek Alliance, and also Climate School at Columbia, and also NSF and Columbia Universities of the programs as well. So this is basically what I will try to presenting, and I'm just to try to give a little bit of advertisement. I recently forming this PASS network. It's called Plastic Pollution Analysis and Sustainable Solution Network. And in that, we actually forming this network that we, forming more than 50 researchers forming this network, so that we coming out from different fields like environmental law, life cycle analysis, environmental impact, new materials, and the environmental toxicity and health, education, etc. So we have all this and like researchers coming together to address these global pollution issues. And we have this bi-weekly seminar, which is roundtable or discussions. And if you want to know more, you can send us the email or visit our website. And this is our email you can go use for that. With that, I will thanks that the invitation from the Hudson River Foundations. I will take any questions. Thanks.
Great. Thank you, Beizhan. That was amazing talk. Super thorough. You covered a lot of ground, and we have a lot of questions. So I've tried to group a few of them, and thought maybe we'd start with sort of thinking about the distribution of microplastics. There was a couple questions on, and this was early on in your talk where you showed Newtown Creek being a major source. So there were two questions looking for your input on why you think that might be.
So I think that in the later talks, I mentioned that the reason is because that Newtown Creek is a long, narrow inlets. And these non-narrow inlets, when they have a CSO, like a lot of CSO, and also there was close to the discharge from Newtown Creek of the wastewater treatment plants. So when they are getting into the Newtown Creek, it's the residence time in there can be the months, or so difficult for them to come out, and so they are also being so that's potentially the reason we think about it, is because coming out from the CSO and also the discharge from the wastewater treatment plants. Yeah, of course that we need a more study to showing the causal effects. So we need a more study, and we can do much more to figure out. Maybe we can pick up the particles and then do the microbial, community work on those things, and then figure out whether they are coming out of sewage. So we're also collecting the samples on the when the discharge side, when the boat comes to over, we're also collecting some samples on this discharge, outfall sites, and then we compare the nearby, a little bit far away, and we see much, higher concentration of microplastics in those sites. So that's basically the sum of the study. Yeah.
Good, thank you. Staying on sort of distribution, Mark Tedesco from EPA. Hey, Mark. His question is, have you assessed the relative contribution of microfibers that are synthetic versus cellulosic, if I said that right? And then he cites a paper. So how you just distinguish between synthetic and natural fibers, I guess.
So the basically that's the kind of when we use the FTIR and also the when we use FTIR and the Raman techniques, we can really identify whether it's coming from natural or not, right? So that's basically using the chemical like a method that we can use. And also the natural ones we typically use that we also use the like H2O2 to remove most of the natural organic matters from our samples. So that's another thing that we are doing to remove that. Yes.
Great.
Yeah.
So next set of questions on behavior in the environment. So question from Bronan Shelby Curran. The question is, how do microplastics and nanoplastics eventually degrade into smaller particles? And do they do that at different rates depending on the type of plastics?
Yeah, that's a great question. So there was a lot of work need to be done on to study that. And so they are, because of the photo degradations, they are making the plastics, easily being break under the, and especially also under the New York City, the, very dynamic, like the New York City, waterways. So they are basically, can breaking into small pieces with time and under the chemical and the biological, degradations. So they are, with time, of course, they're getting into smaller pieces. And so, but they are still have a lot of research need to be done. And in terms of different type of the plastics, and that's a really good questions. And I didn't do the research on that, but I do know some of the research that researchers to compare the different type of under the, like accelerated weather conditions, how quickly of the rates of the degradations, also maybe the fragmentations. And I don't have anything in my research doing that. So they will do some of the work, and I can send these results to if they was asking me, send me email. Yeah.
Yeah, no, that's great. So I think you just answered it, but Elaine Thruner also had a question on if microplastics would be affected by water temperature, and specifically like thinking about surface ice or flooding or ice jams and things like that.
For the ice, when they are forming the ice, right? So of course, when they are forming ice, probably the plastic was being entrapped inside of the ice and then being, like moving around with the ice. And they do see that, like high concentrations of the well, I will not say the high concentration, but they do find out that microplastics even in the polar region in the Antarctic Sea, like ice in there, and showing that it's actually entrapped inside of the ice, like structures. Yeah.
Okay. Switching to there's several questions about methods, and some of these I think were answered later. So maybe I'll jump to and maybe I'll encourage people to also reach out to you directly because some of these are quite specific. But there were two related to the Raman microscopy and the use of the Nile red and whether that technique could be applied to like using FTIR as well.
Yeah, of course. That so the Nile red and the problem. So for we actually doing the experiments in a serious ways. So we before they do the Nile red, we actually doing the FTIR and Raman, and then we do the Nile red. And so because the Nile red is easier to identify and then get the samples counted. However, for the identification for the larger particles, we actually using the FTIR and Raman. And so the the method we are doing right now, which is like as I mentioned, we have developed these advanced techniques with my colleague at the chemistry department. We use these stimulated Raman techniques that it's being the first time being used for the identify nanoparticles, and we actually being able to identify those nanoparticles down to like 100 nanometers, and we did identify that in my like laundry waste samples. You can see that was a lot of tiny, less than 0.6 microns. They are actually using that techniques. We also identify in the drain, in the drinks like soda, and also in the bottle of the waters, and also in many other like samples. So the one paper was actually in the revisions. We submitted this paper to the PNAS, and so right now it's in the revision. So looks like within probably 2 months we should get that published in the PNAS. Yeah, to reporting our findings. Yeah.
Great, thanks. And there's a couple questions on the modeling. Well, there was a comment that amazing simulations. So I agree with that one. It's
thank you, Phil.
Thanks, Phil. And so, Phil, you're welcome to come off mute if you want to answer this. But there's a question from Robin Miller on how you're defining residence time, and specifically, how wide an area are you releasing the particles into, and when do you, in your display, when are you when do you stop showing it, I guess, like, is it at a, there's absolutely no particles left? And then I'll add in my own sort of question around the same lines is, what are you modeling different particle types when you're doing the residence times? And if you're not, would you expect the model, I would expect it to be sensitive to particle size and different types. And so maybe you can comment on that as well.
To the first question answers, and I did answer these. I typed in an answer too if anyone needs to look back. Their point source releases in the model, in both cases at the bottom, in East River, and then also, which is where the wastewater treatment plant outfall is, off, out in the middle of East River near Newtown Creek, and then off North River treatment plant along the Hudson. The time it takes, the residence time is defined as the time it takes to decrease down to 37% of what was initially put in. It gets injected over 12.4 hours just to capture what would come out in one particular tidal cycle, but it's always coming out, these microplastics. And then the residence time was defined as that decrease to down to 37% of what the original amount was. If you want to think about how long it would take to decrease down to 14%, that would be double that time. It's called an E-folding time, and that would be the amount of time it would take to decrease by about a factor of 8. So you just take double the residence time. The residence time is defined as the whole harbor region, and there was a map shown. It goes up to northern Haverstraw Bay and out to Ambrose Channel and out to Willets Point in Long Island Sound. It includes Newark Bay. So it's sort of a big harbor area, kind of an arbitrary region. And then to answer Jim's question, this is pretty simplistic. It's just neutrally buoyant tracer. It's just something that follows the water. Some of the simplifications are it won't capture buoyant particles as well, unless they're really being stirred around by turbulence enough that it doesn't really matter that they're buoyant. But so it's better for certain. Maybe Bay can follow me and mention which particles he thinks it best accounts for. But a lot of the finer particles, or the particles near a density of 1, would be captured. And then it does neglect whether or not they get, I don't know, and maybe Bay can also comment on this, they may eventually, instead of their fate just being governed by the water flow, it might also get governed by association with particles of other things like mud and sediment. If there's attractions and they form into agglomerates of different things, then that could raise the particle settling rates, and maybe they would settle out. So that's another simplification.
Great, thanks.
Yeah, exactly. So if you have like a biofilm forming in there, and also when you have invertebrates that coating on the surface, then the density will change over the time because of the biofilm and also the other materials being on top of it. So that's why of course this is needed to be much more work, and this is like a simple model, but did give us a time like 10 days to 33 days that actually giving us our estimation of the residence time in them. Yeah.
Great. Well, I think we're right about 12 o'clock. I just want to thank you again, Beizhan and Phil. I think really amazing talk. I certainly learned a lot. I think others did as well. And so thank you very much. And everyone, thanks for joining.
Okay, thank you very much. Thanks for the invitation. Yeah. Okay. Bye-bye.
Bye all.